Good morning to all. I welcome you all for this National Science Day 2024, organized by School of Life Sciences, B.S. Abdur Rahman Crescent Institute of Science and Technology, in association with the Tamil Nadu State Council for Science and Technology, Government of India. So my topic is an emerging pathogen, which is an
Good
Vibrio
morning to
fluvalis.
everybody. This
As
is
you all
Dr.
aware of
Baskaran. I'm presenting
cholera
the most
caused by
important
Vibrio
organism called
cholerae,
Vibrio
which
fluvalis,
is a pathogen
which is an
causing a
emerging
diarrheal
pathogen
infection.
in recent
It
days.
causes more than
So,
eight
however, it
episodes
is a
of cholera
neglected
outbreaks, which
pathogen
we
which
are all
is
know.
not
But the most neglected
consolidated and it
pathogen,
is not
where
done by
most
most of the
people
work.
were not focusing, which is recently an emerging pathogen, which is said to be as Vibrio fluvalis. Let's see about that emerging pathogen, Vibrio fluvalis. Uh, Coming to introduction, diarrheal diseases like cholera, gastroenteritis are the major problem in countries with poor socio-economic condition where access to safe water and adequate sanitation cannot be assured. So, in order to maintain the proper sanitation, we should regularly monitor the presence of these diarrheal pathogens in most of the food and water, both as well as food as well as in water. Nearly 3 to 5 million cholera cases have been reported by Center for Disease Control. It has been reported that nearly 857 deaths were reported throughout the countries. It is an WHO report. So during the 19th century, cholera spread across the world. First pandemic truly originated from Ganges in India. So technically, this outbreak, diarrheal infection, which is a cholera outbreak, it was first reported in India only. Later on, there will be several episodes of cholera. Subsequently, several pandemics which killed millions and millions of people across the continent. So, like more than 8 episodes of cholera outbreaks have been reported. Let's see one by one. Vibrio fluvalis. It is a newly emerging and neglected human pathogen that causes gastroenteritis, and these pathogens, Vibrio fluvalis, found to be associated with marine environment, which means seafoods and sea-associated foods. Those things they found to be associated in marine and marine organisms. So technically, it causes similar to kind of Vibrio cholerae, which is a cholera disease. So similar to cholera like bloody diarrhea, it also causes wound infection and septicemia. In 2009, it was reported in Calcutta, Belgeta ICMR Institute. They reported that nearly 131 pathogens were out of 131, 43 were sole pathogen and 88 were co-pathogen, which means 43 were Vibrio fluvalis and 83 were co-pathogen, which means this Vibrio fluvalis was found to be associated with other Vibrio. Say, for an example, if you're isolating some sample from diarrheal patients, not only Vibrio fluvalis, you found to be other pathogens, including like Vibrio cholerae. This is they reported that this was associated with other pathogens, Vibrio cholerae, Vibrio parahemolytica, sometimes Salmonella. So, what are those virulence factors? So, what causes infection in humans? Uh, those things are cytolysine, heat level cytotoxin, Elta like hemolysin. So, Elta like hemolysin, it's technically from classical. It's a differentiation based on uh, lysed with chick embryocytes and also with the agglutination with the polymyxin O. So based on some biotypes, will classify organism either it is a classical type or ELTA type. So it shows type like hemolysin. So technically, it will be confused with Vibrio cholerae and Aeromonas. So if you isolate the organism from seafoods or diarrheal patients, you will come to conclude that it is a Vibrio cholerae or Aeromonas. But technically, it is not like that. So it is a different species, Vibrio fluvalis. Ecological and molecular epidemiological studies of this particular pathogen is remain unclear. So there is no report on this particular pathogens. Still, most of the research is going on. Say, for an example, National Institute of Cholera and Enteric Diseases, Dr. Chakraborty and various research groups, they are focusing on this particular pathogen. We will see the remaining things in next slide. How to isolate and identify this Vibrio fluvalis? What makes difference or what is the difference between other Vibrios? This picture shows like this is how fish market when we go here in Chennai, not only in Chennai, throughout the Tamil Nadu, wherever you go, it will be like that. They use the same water, which means fish washed water throughout the whatever they are doing. They use the same water, which means they are not maintaining proper sanitation. They are not maintaining cleanliness in those environment. This is what where we are buying most of the seafoods. This is the different pictures which was taken from fish market in and around Chennai. Uh, How to isolate these organisms? Say, for an example, if you are collecting seafood samples or like environmental samples, so there should be very concern about the samples, whether you are isolating from environmental samples or clinical samples. Though the organism is Vibrio cholerae, Vibrio fluvalis, the protocol varies from sample to sample, which means if you're isolating any seafoods, technically, it should be enriched with alkaline peptone water and then plated on thiosulfate citrate bile salt sucrose agar, followed by you will be saw green colonies. You should be focusing on isolating these colonies. It resembles like same like Vibrio cholerae. So technically, it is like a yellow color colonies, similar with that phenotype. If you plate it on TCBS, however, you have to go through a series of biochemical reactions to confirm or to differentiate. It is an Vibrio cholerae or Vibrio fluvialis. Say for an example, you have to go through IMVIC test followed by lysine decarboxylase, ornithine decarboxylase, arginine dihydrolase, and L-arabinose, those amino acid tests to confirm whether it is an fluvalis or other species. However, these fluvalis can be confused with Aeromonas as well, which means it shows the same colony morphology, same biochemical reaction. However, you have to go through a serotyping technique to confirm it is an woven or non-woven. Technically, Vibrio fluvalis, it is a non-woven serotypes. These are the different identification technique used to confirm Vibrio fluvalis, or otherwise used to differentiate Vibrio fluvalis from other Vibrio species. Those methods are fast typing, which shows like 73 percentage identification, 16S, 23S intergenic spacer, IGS sequence, bacterial 16S rRNA, conserved transcriptional activation and variable membrane tether region, tox or regulatory genes in Vibrio fluvalis, other hemolysin genes or heme utilizing genes, central regulation genes, as well as multiple primer extension reaction assays, pyrolysin mass spec, multiplex PCR. These are the different techniques were adapted by different research groups. If you ask me which is the best method to confirm Vibrio fluvalis, I would say this transcriptional activation regulator, toxo regulator for identification of Vibrio fluvalis is the best method. Uh, uh, though there are several methods, you want to make sure using other techniques also. Say, for example, whole genome sequencing or a pulse field gel electrophoresis or mismatch amplification mutation assay PCR, those things. Coming to phenotypic and genotypic characteristics. Coming to phenotypic and genotypic characteristics of Vibrio fluvalis, you can confirm through serotyping, say for an example, flagellar antigen, and also like somatic antigen and H flagellar antigen. Technically, it is not in practice, though other species, though other strains like Vibrio cholerae and parahemolyticus, that is technically used for serotyping, whereas this species, fluvalis, there is no proper technique for identifying those serotypes. However, molecular typing methods like pulsed field gel electrophoresis has been proven highly useful in typing Vibrio fluvalis, which shows gene diversity of that particular organisms. It was if it is collected from both environmental as well as clinical samples. Coming to prevalence of Vibrio fluvalis from marine environment, depending upon the conditions like physiochemical properties and physicochemical conditions and biological parameters, it found to be associated with various marine environments. It was found to be predominant and it is detected in soil sediments, several environments, both in summer as well as in winter months. Other countries like Florida, USA, Toulon, France. It found to be associated with other organisms like Vibrio cholerae, Vibrio parahemolyticus, Vibrio vulnificus, and Vibrio mimicus. Predominantly, it is isolated in river water, soil, sediments, plants, estuaries, plankton blooms, zooplankton, marine mollusks, bivalves, oyster, and various seafoods. In India, the distribution of this particular pathogen was reported in Ganges, nearly 0.74 percentage, Varanasi and Myanmar. Recent study showed that Atlantic coast of France showed based on chlorophyll A pigment influence that also influence the growth of this particular pathogen Vibrio fluvalis, which is isolated from mussels. Coming to sporadic cases and outbreaks, since 1978, Vibrio fluvalis were found to be associated with gastroenteritis in USA and Japan. Later on in 1988, incidence of fluvalis among children also causes acute diarrheal infections in individuals, which has been reported in Calcutta and also in China. But in China, it has been stated that second most pathogen when comparing to Vibrio parahemolyticus. And in 1996 to 1998, nearly 9.4 percentage of hospitalized diarrheal patients in North Jakarta was due to Vibrio fluvalis infection. Subsequent foodborne diarrheal outbreaks have been reported in Maharashtra in 1981 and as well as in Calcutta 2012. In Volga Delta, Russian, it causes acute enteric infection, which was reported by Vibrio fluvalis, isolated from sea and freshwater fishes. When comparing to traveler's diarrhea, this organisms was very low when comparing to other enteric diarrheal pathogens. Coming to virulence factor of Vibrio fluvalis, it produces, as I said, it produces delta-like hemolysin, which can be confirmed by sheep blood agar plate. Technically, it will produce hemolysin. And then, apart from this, it has in vitro cytotoxicity production. When you use the Chinese hamster ovary cell lines, it also has killing factors, cytolysin against RBCs, protease active against azocasin. It also produces heat level cytotoxin, which is a major or putative virulence determinant of this particular pathogen. These are the other virulence factors. Say, for an example, cytolysin, heat level cytotoxin, cytotonic, hemolysin, mucinase, manosensitive hemolysin, hemoagglutination, and then cell adherence, cell vacuation. These are the other factors which was associated with Vibrio fluvalis. This particular organism even has the capacity to survive in seawater microcosm for more than 15 days with ambient temperature, regardless of the carbonated substrate. And moreover, it has been found that these organisms enter into a non-culturable state, which is otherwise said to be as viable non-culturable state. And it has been found that it could be resuscitated with supplemented with nutritional requirements for that particular organisms. Antimicrobial resistant of this particular pathogen. They are found to resistant like ampicillin, carbapenicillin, kanamycin, ceftolozin, trimethoprim, chloramphenicol, ciprofloxacin, and polymyxin B. Several mobile genetic elements, otherwise said to be as jumping genes, which carries these antimicrobial resistance, found to be present in Vibrio fluvalis genome. The integrative and conjugative elements which is a transposon commonly detected in Vibrio cholerae was also found in Vibrio fluvalis. Technically, it carries the resistant genes for sulfamycoxyl trimethoprim, which is SXT gene elements, chloramphenicol, and streptomycin. The aminoglycoside acetyltransferase encoding genes was also identified in class 1 integrons from clinical samples. Uh, Two putative multi-antimicrobial extrusion protein family that affects pumps through H&D type were responsible for fluorochemical resistance were found in Vibrio fluvalis as well. Most of fluvalis strain has mutation which is seronin to isoleucine at the position of 83rd of the equivalent resistant determinant region of gyrase A at the position 83 and PAR-C at the position 85. Plasmid-borne clonal resistant gene was also reported in Vibrio fluvalis. In addition, these strains carries transferable 150 kb base pair plasmid that harbor the clonal resistance, which is a complex sulfur mixes in type 1 integron. These resistance have been found in Vibrio fluvalis, which has been reported by various research groups. Though the pathogen Vibrio fluvalis has known for quite some time, its clinical importance is now realized that the prevalence of diarrheal cases is reportedly it seems to be increasing in recent days. In-depth studies on or understanding the pathogenesis of this particular Vibrio fluvalis, it has to be established as there are many description about these virulence, putative virulence factor in general, food, seafoods in particular, and also like food as well as water, found to be the major reservoir in transmitting these type of infection among individuals. So technically, controlling diarrhea infection lies in simple hygienic measures that aimed not only preventing the multiplication of these emerging pathogens in food as well as in water, but also helps in reducing the transmission of this pathogen in contaminated food as well as in water. Thank you so much for listening this presentation. Please let me know if you have any questions. Thank you.